Okay. So I'm the founder and CEO of Prellis Biologics. I was part of IndieBio's Class 5. I have a PhD in physiology and biophysics, where I spent my time studying the immune system as well as additional tissues within the immune system. The immune system is actually extensive. It's throughout our body. So gave me a really good background to start a tissue engineering company. I've spent 15 years looking at pretty much every tissue in the body. And while we were part of Class 5 at IndieBio, one of the first tissues we built as a tissue engineering company is something we call mini human lymph node. It's basically an externalized human immune system that you can use to test viral responses. You can use it to make antibodies, and that's what we did. And so when this crisis became apparent that it was going to become a global pandemic, we took a hard look at restarting that technology, which we have granted IP on, and decided to shift our efforts towards producing monoclonal antibodies that are virus neutralizing. And I'm happy to share what we're doing and a progress update there. So because we can do this with an externalized human immune system, we get the immune cells from a blood draw, we can produce these neutralizing antibodies in about 30 days in the lab. So we're faster than any other technology out there. It often takes people several months to get this going, even if they're getting the cells from someone who is already infected. So happy to walk you through the process. So this is my thinking. In about 3 months, our world was shifted forever. And so it's time to do something about it. This is the greatest pandemic in living memory. And my opinion, as someone who studied immunology for many years, is that this is not going away. It's going to be persistent. And we have produced pandemic-stopping antibodies in the lab now, and we're currently screening them. So I'm really happy to share why and how we're doing that. So as I said before, we produce living human tissues. We can leverage these human tissues to produce valuable products such as antibodies. Human albumin is another one. And this ex vivo human biology can produce products that are made outside of the human body in a really safe, quick way. So why antibodies? We consider them a modern panacea. Human antibodies are highly specific protein tools that our immune system rapidly develops to stop pathogens in their track. They can completely neutralize the pathogen, and just a few hundred milligrams can protect you from viral or bacterial infection. And it is widespread in clinical use. And human antibodies from a human source are generally considered safe. And so we believe that we'll be able to move through an FDA approval process very fast with this. So why produce antibodies? Again, they confer passive immunity for up to 3 months. And they can be used repeatedly in patients without side effects. So they have a pretty good dosing profile as long as there's no additional side effects associated with them. This virus is starting to undergo antigenic drift, and I'll share some of the information on that with you guys in a minute. So that means that prior vaccines that take a year and a half or more to develop, antibodies from convalescent serum or from other sources are going to be quickly outdated as this virus evolves. You can, using human antibodies, you can get some rejection of the human antibody in patients. So having backup antibodies is really important. You want to have more than one shot on goal in a situation like this. There has been some evidence that coronavirus immune responses are not long-lived, and this is from papers looking at the SARS-CoV from 2002, 2003. A lot of patients about a year and a half, 2 years later have lost their protective antibodies in the system. And so having something that we can inject therapeutically to take that place is a good strategy in my mind. And genetic diversity of human antibody production is also something that could be very important. By just taking a simple blood draw and then growing up hundreds of these in a dish, we're able to parallelize development of multiple people. And so we can really leverage genetic diversity and search out the best antibodies cheaply and quite efficiently. And we do have the potential to start creating a universal antiviral cocktail where it doesn't matter what strain of the virus you have, we could have 5 or 6 antibodies injected together that would neutralize the virus on site or confer protective immunity for frontline workers or physicians. And so we are quite novel in the way we do this. It doesn't require human infection. We don't have an intensive screening process. It's actually quite fast. We developed our own in-house ELISAs for this specifically. We can do it in about 4 to 5 weeks, as I mentioned before. Other methods of getting after human antibodies, especially with people who've been infected, include a lot of travel time, labor, patient contact, in silico antibody development. Doesn't guarantee good hits. And then it also is quite labor intensive in the screening process. So we believe we've kind of hit that sweet spot of leveraging human tissues to do what they do best, to get after the best possible antibodies. And so we've done this before. Like I said, in 2017, we were part of IndieBio's Class 5, and we produced human antibodies against the Zika virus. This is data that we saw, you can see over there, well D3. Had stronger antibody responses than the kit's positive control. So we believe we have a very good antibody there. And we moved on from this, though, because we weren't set on becoming a therapeutics company. We're really a tissue engineering company. That said, though, we did file IP on this, and we did have a patent granted in late December of 2019. And so here's how the process works. We start with the heat-killed virus or gamma-irradiated virus. We've already obtained that from the CDC, and we've been using it in the lab. We have our mini human lymph nodes. We create them, and then we inoculate them, much like you would give someone a vaccine shot. We basically make our own vaccine cocktail in the lab. And then we do monoclonal antibody screening from those, and then we create long-lived hybridomas. We fuse the antibody-producing B cell with a myeloma cell. This technology has been around since the 70s, and it continues on as the cell line that just spits out the antibody, and we can go into sequencing, mass production from there, GMP scale-up and distribution. So currently the company is right here. We're screening our final hybridoma clones. We have over 50 human antibodies that bind to the S1 or the S2 of SARS-CoV-2, the Wuhan strain. And we're quite happy with that. And we expect to be making some announcements about that in a couple of weeks. So you guys get to hear it here first. And so we really did leap into action around this. We shifted our whole lab process in 1 week. We obtained the virus from the CDC. We reached out to other antibodies. We re-engaged UCSF to blood bank to get blood. We became a BSL 2+ certified laboratory, and we started recruiting people to work on this. And one thing that's really personally exciting about this technology for me is that we can actually beat the viral mutations. As fast as they're discovered, we can get an antibody into our production pipeline and actually perhaps isolate and stop the spread of specific strains that other therapeutics have lost efficacy to. So we can beat the virus, and then we can actually start to a priori predict mutations and make antibodies to those proteins. And because it's so inexpensive for us to do this, we could have those in stock on the shelf and ready to go. All right. So this is very current. This is actually a slide I put together last night with about 40, maybe more like 15 represented here, but there's about 40 or so amino acid mutations that have been identified in the virus, and it seems to be regional. This means, unfortunately, that I think it's going to become more of a seasonal virus. There hasn't been selective pressure demonstrated to make this a less virulent virus than one that doesn't spread. And so having technologies that can adapt quickly to this, and rapid testing technologies as well, really will help. One interesting point I just highlighted it in red, is that we do have this mutation D614G, and it does seem to be a super spreader virus. It took over the US viral population in about 2 weeks. It came here from Europe, and now everyone who's testing positive in the New York area and moving west is showing up with this virus. It doesn't seem to have a lower death rate associated with it, which is too bad, but it does seem to have a higher spread rate. And the other thing that's interesting about this chart too is that a lot of these viruses are located in specific areas, and they aren't spreading outside of them. And so that means that we're going to start to have isolated virus mutation in specific areas where they accumulate mutations, as we have ceased most of our global travel. Okay, so other solutions. There are other solutions to this that are very similar, and I've mentioned or briefly touched on them before. You can get B
Actually,
cells.
Melanie,
Oh, are we almost done? Okay. All right. So I'll just
yeah,
introduce my team.
we didn't need that. Yeah, you're great.
Sorry. Yeah, so I'm just really proud of my senior team here. They're all highly experienced and many, many PhDs on our team working with us, prior founders, and we're hoping to move this to market as fast as we possibly can. So thank you, everyone.